Hello everyone. So I'm going to jump through the beginning of this kind of quickly because it's stuff we've already talked about. But I'll just kind of hit the highlights as reminders and then focus on cell transport because that's kind of the newer stuff. So memory and function. Remember we're talking about life at the edge of the cell, and for today, the most important thing is going to be that it's selectively permeable. So some things can go across the membrane and some things cannot. So you see the structure there. Notice the proteins that are embedded. They can act as channels, which we'll talk about today. It's a fluid mosaic model. So it's not static, but it's both sort of moving and changing all the time. Okay, I just want to remind you one more time of the two parts of the phospholipid bilayer. So this top part represents all of these little purple dots. Those are the phosphate heads. They are hydrophilic. The phospho part is hydrophilic. Then the lipid tails are hydrophobic. So they make kind of that lipid sandwich in the middle. Okay, so I'm going to jump a little bit. Remember, you have all these notes available to you on Schoology. So look at them. So this we talked about, I think, in both classes already, but just I'll remind you. The lipid bilayer, remember, it's semi-permeable. Small hydrophobic molecules, since they're nonpolar, they can go right through the membrane, carbon dioxide and oxygen, for instance. Small polar molecules can squeeze across the membrane. Water and glucose, since they're small, they can kind of fit through the spaces. Things that are charged can't cross the membrane without help because they can't make it through that nonpolar environment. And then big molecules need either a protein to help them, or they will cross the membrane in a different, using a different mechanism. So, as you know, I imagine, because we've talked about it many times before, there are certain things that cells have to do in order to survive. So they have to metabolize, take food and turn it into energy. They have to exchange carbon dioxide and oxygen. They have to divide. They have to be able to communicate. And so all of those things take place through importing and exporting across the cell membrane. And the fact that it's a selectively permeable membrane is going to sort of determine how things move across the membrane and also how that is regulated. So it's a very complex process. We're going to kind of skim the surface of it. So there are three main ways for molecules to move in and out of the cell. Passive transport happens just sort of with no input of energy. It's just what naturally happens. It's a diffusion of a substance with no energy investment. Facilitated diffusion is still diffusion, so it's still kind of what happens naturally from high concentration to low concentration, but there has to be a protein channel for that particle to go through. So it can't squeeze through the membrane, the sort of sandwich of the membrane, but it can make it through. And then active transport is active in the sense that it requires energy. So it requires ATP, which is our energy molecule, to move across the cell. And this can happen in two ways. One is on the molecular scale, So when just maybe ions are being pumped across the membrane, or in a bulk transport. So like cell eating and cell drinking. So diffusion, you all did the demos, and you saw how the food coloring sort of started in one small spot when you added it to the water, and then it spread throughout the whole beaker more quickly or less quickly depending on the situation. You also hopefully saw that with the smelly balloon, the gas molecules of the extract sort of permeated the room from high concentration to low concentration. And we call that down the gradient, down the concentration gradient. So from high to low, you're going down, sort of down the hill. And you can, if you want to Google, you can find, I'm sure, a lot of illustrations or animations that show sort of how the molecules diffuse. So these green molecules here would eventually just be all spread out throughout the room, or the whatever the blue is, the water. Another important thing is that it's a dynamic equilibrium. So it's not as though the molecules spread out and then are just stuck in place. They spread out and then they're still moving around. They're just moving sort of equally so that they're remaining all spread out throughout this space, the room or the beaker or whatever. But movement doesn't stop. It keeps going. Osmosis is a specific kind of diffusion that occurs with water. So I never ever want to hear you studying by osmosis unless you're studying in a pool. You can study by diffusion if you prefer. So when we're talking about a solution, remember we're talking about a liquid solvent, often water, and then the solute is the solid. Sometimes it'll be sugar or starch or something. And when we compare solutions, when we're looking at how the water is going to move, we talk about what's called tonicity. And you saw this with the dialysis bags in the demos. So something that is hypertonic is going to have a higher solute and less water. Something hypotonic is going to have a lower solute and more water. And then isotonic is about the same concentration. So if you think about the word iso, like isosceles triangle, the sides are the same. So isotonic, it's the same. If you think about hyper, like if you are hyper, you have more energy, so there's more solute. So here's a couple of examples of that. If you put an animal cell, remember animal cells, they have cell membranes, but they don't have cell walls. They're just being held together by that membrane. If you put a cell which has lots of stuff in it, lots of solute, right, in a hypotonic solution, if you put it in pure water, then the water will rush into the cell. It tries to dilute all that solute, tries to diffuse in, and the cell bursts. If you put it in an isotonic solution, the water going in and the water going out are about equal. If you've ever gotten an IV at the hospital, they have to make sure it's an isotonic solution so that your cells are kept safe while getting the IV. And then if you were to put a blood cell in a hypertonic solution, so this would be like very salty water. Very salty. The water inside the cell would try to dilute that very salty water. And so the water would rush out and you get this sort of very shriveled cell. So basically, whenever you're looking at a tonicity scenario, the water is going to move to wherever there's more solute. Okay, so if there's more solute inside the cell, it goes in. If there's more solute outside the cell, it goes out. It's just trying to dilute the solute, basically. Paramecia are animal-like protists, so they don't have a cell wall. They live in fresh water, and so fresh water is constantly moving in to try to sort of dilute the contents of the cell. So they could end up bursting, just like we saw that first red blood cell. But they have an adaptation to allow them to maintain homeostasis. So they have a special organelle, it's called a contractile vacuole, and it contracts and squeezes excess water out from the cell to help it maintain the pressure that it needs to have so it doesn't burst. This is another thing I encourage you to Google and see if you can find a little video of this. I'll see if I can find one too and post it on Schoology because it's pretty cool. Plant cells, since they have cell walls, they don't have the same problem with bursting that animal cells do because the plant cell wall holds sort of the membrane in place. It gives it protection. And so plant cells actually are happiest in a hypotonic environment. So you see that right here. Hypotonic environment. So this is like pure water again. And so the water rushes into the cell, just like in the animal cell, and the cell membrane pushes out against the cell wall, and that actually helps give the plant structure. Here we have, this is the isotonic solution, and this would be like a wilty kind of a plant because there's no pressure pushing out against that cell wall. Cancel. And then this last one, this would be salty water. And you can see that the cell membrane has pulled inward away from the cell wall. It's gotten all shrunken because you don't have that fresh water pushing in. Water instead is rushing out of the cell, trying to dilute the outside environment. So that was diffusion, regular diffusion. Facilitated diffusion happens when you have often something bigger that can't go through the cell membrane easily, and they allow one specific molecule to cross the membrane more quickly than it would if it just had to squeeze across the membrane. It's still going, you can see, from high concentration to low concentration. So it's still just going down the concentration gradient. It's just happening through this channel protein, a transport protein. It's like having a special tunnel to pass through, like VIP status or something. So both of those transports that we just talked about require no energy. They just happen naturally because that's how things move naturally. Second law of thermodynamics. Often in the body, though, things need to move the other direction. They need to move from low concentration to high concentration to kind of build up a supply on one side of the cell. To do that, since it's sort of the unnatural way to do it, the non-physics way, it does require energy, and usually the energy is in the form of ATP, which is what gets made in the mitochondria from sugar. So a famous example, and please don't pay attention to this page in the book, but it is section 7-3. A famous example is the sodium-potassium pump. Your nerves work by sending sodiums into the cell and potassiums out of the cell and that's kind of what propagates the signal. And so in order for that to work, your body has to build up a big supply of sodiums outside the cell and a big supply of potassiums inside the cell. And so you have these special pumps called sodium-potassium pumps, cleverly, that pump sodium out, okay, from low concentration to high concentration, which is weird. And then they pump the potassium in from low concentration to high concentration. So they're setting up a gradient that can then be used later on to send signals through your nerves. So it's using ATP all the time when it's pumping because it's trying to do work against the concentration gradient. Bulk transport is another way of moving things. It does require energy, but it's a good way of moving a large quantity of stuff all at once. And that can either occur into the cell or out of the cell. Endocytosis, bringing things in. So the cell is going to form like a little bubble, a little vacuole around whatever macromolecules and pull it in. And exocytosis is kind of the opposite thing happening. A little bubble, maybe from the Golgi, the little vacuole, moves out to the cell membrane, fuses with the cell membrane, and then releases all of its contents. So here's an example. This is an amoeba. It's a single-celled protist, and they eat, which is what phagocytosis is, just cell eating. It's a type of endocytosis. And so you can see here, the cell is coming around, and it's going to eventually make a complete circle, just like it did here, around this food particle. And then it will bring it in, and then package it into a little vacuole. Maybe it will send a lysosome over to combine with that vacuole and digest whatever this is. And that's, you can imagine that that's what these things are too. It's food that's going to be digested. So it's kind of cool. Here's another example. This is a macrophage. So this is a white blood cell that's going to eat microbes and sort of break them down in order that they can be presented to the immune system. So the macrophage will come, it will eat the microbe, then it'll travel in, see it's in a little vesicle. Here comes the lysosome to break it down, and then you get sort of this leftover junk that then gets expelled. So this little vesicle is going to bond with the cell membrane and then get released. So this would be endocytosis on this side, number 1. And then this down here at number 7 would be exocytosis. In and out. So here's another picture of exocytosis. And this can be a little reminder of cell organelles that we've talked about. So here's the nucleus. Here's the rough endoplasmic reticulum. It's sending out a little bundle of proteins. Maybe it's insulin in the pancreas. Okay, it gets sent to the Golgi. Remember, the Golgi sort of adds things to it if need be, or modifies it in some way, and then they can mark it for delivery. So if this is some insulin, it's going to be marked for exocytosis. And so it gets sent out to the cell membrane, And this little bubble here is going to fuse with the cell membrane, release its contents, and then it will just become a new part of the cell membrane. And that is it. Diffusion, facilitated diffusion, which require no energy, and then active transport, either as like a sodium-potassium pump, or exo and endocytosis. Those require energy. So, read in the book, section 7-3, to help you review as well. Bye!